There is a molecule inside every living cell on Earth that contains the complete instructions for building and operating the organism that cell belongs to. In a human cell, this molecule, compressed into a space so small that 100,000 of them stacked on top of each other would be no thicker than a sheet of paper, contains approximately 3 billion chemical letters arranged in a specific sequence that spells out the complete recipe for a human being. Every protein the body makes. Every enzyme that drives every chemical reaction. Every structural component of every tissue. Every instruction for when to grow and when to stop growing and when to repair and when to die. All of it encoded in a sequence of 4 chemical bases, adenine, thymine, guanine, and cytosine, arranged along 2 intertwined strands of a molecule whose specific geometry is one of the most elegant structures in the history of biochemistry. The double helix of deoxyribonucleic acid. DNA. The story of its discovery, of how human beings came to understand the structure and the function of the molecule that carries the code of life, is one of the most dramatic, most contested, and most humanly complex stories in the entire history of science. It involves a sequence of brilliant minds building on each other's work across nearly a century. It involves the specific social injustice of a woman whose crucial contribution was used without her knowledge or credit and who died before the Nobel Prize she should have shared was awarded. It involves a race between competing laboratories whose competitive urgency produced one of the most famous eureka moments in scientific history. And it involves the specific conceptual leap, the recognition that the three-dimensional structure of a molecule could itself encode biological information, that changed everything about how humanity understood the nature of life. Before the double helix. Before Watson and Crick and the model made of tin plates and wire in a Cambridge laboratory in February 1953. Before Rosalind Franklin's X-ray photographs and Erwin Chargaff's base ratios and Linus Pauling's alpha helix. Before all of it, there was the long and difficult road of establishing that DNA was the molecule of heredity at all, and before that, the even longer road of establishing that heredity had a molecular basis in the first place. The concept of heredity, the specific transmission of biological characteristics from parent to offspring, had been observed and puzzled over since the beginning of human engagement with plants and animals. The farmers who selectively bred their livestock, the horticulturalists who developed new varieties of flowers, the physicians who noted that certain diseases ran in families, all were observing the phenomenon of heredity without having any conceptual framework for understanding its mechanism. Gregor Mendel, the Austrian monk who conducted his famous experiments with pea plants in the monastery garden at Brunn in the 1860s, established the mathematical laws of inheritance that provided the first rigorous description of how characteristics were transmitted from parents to offspring. His careful statistical analysis of the inheritance of specific traits in thousands of pea plants, the color of the flowers, the texture of the seeds, the height of the plants, revealed the specific pattern of dominant and recessive inheritance that is the foundation of classical genetics. Mendel's factors, the units of hereditary information that determined each trait, were the conceptual ancestors of what we now call genes. Mendel published his results in 1866 and they were ignored for 35 years. One of the most famous cases of premature scientific publication in the history of biology. When his work was rediscovered in 1900, simultaneously by 3 independent researchers who had reached similar conclusions and then found Mendel's earlier publication in the literature, the field of genetics was born and the search for the physical basis of Mendel's hereditary factors, the search for the molecule of heredity, began in earnest. The discovery of the cell nucleus in the 19th century, and the specific observation that the chromosomes within the nucleus behaved in cell division in exactly the way that Mendel's hereditary factors were predicted to behave, established that the chromosomes were the physical carriers of genetic information. The chromosomes were made of protein and a substance called nucleic acid, and the question of which of these 2 components was the actual carrier of hereditary information would not be definitively answered until 1944. The nucleic acids had been discovered by Friedrich Mies-Scher, a young Swiss biochemist who was studying the contents of white blood cells in 1869 when he found, in the nuclei of the cells, a substance rich in phosphorus that behaved differently from any protein he had encountered. He called it nuclein, and in doing so identified, without understanding its significance, the molecule that would turn out to be the most important in the history of biology. He was 25 years old. He had no idea what he had found. For most of the subsequent 75 years, the scientific consensus held that proteins, not nucleic acids, were the carriers of genetic information. The reasoning was straightforward and ultimately wrong. Proteins were known to be enormously diverse, with 20 different amino acids capable of being arranged in an unlimited variety of sequences, making them candidates for encoding the enormous information content that heredity required. DNA, by contrast, was known to contain only 4 different bases and was assumed by many biochemists to have a repetitive structure that could not encode significant information. The specific experiment that overturned this consensus was conducted by Oswald Avery, Colin MacLeod, and Macklin McCarty at the Rockefeller Institute in New York in the early 1940s. Their experiment was based on a phenomenon discovered by Frederick Griffith in 1928, the transformation of pneumococcal bacteria, in which the genetic characteristics of one strain could be transferred to another by exposing the second strain to material from the dead cells of the first. Griffith had called the transforming substance the transforming principle without being able to identify it. Avery, MacLeod, and McCarty spent years systematically identifying the transforming principle, treating the transforming material with enzymes that destroyed proteins, RNA, lipids, and polysaccharides in turn, and demonstrating that none of these treatments destroyed the transforming activity. Only one treatment destroyed it. Treatment with deoxyribonuclease, the enzyme that specifically breaks down DNA. The transforming principle was DNA. Published in 1944, this was the first definitive demonstration that DNA, not protein, was the molecule of heredity. The 1944 paper was received with considerably more skepticism than it deserved. The identification of a complex, apparently repetitive molecule as the carrier of the enormous information content of heredity seemed implausible to many leading biochemists of the period. Avery never received the Nobel Prize for what was arguably the most important biochemical discovery of the 20th century. He died in 1955, 2 years after Watson and Crick's structure paper, without the recognition that the significance of his work demanded. Erwin Chargaff, an Austrian Jewish biochemist who had fled Nazi Germany and joined Columbia University, read Avery's 1944 paper and decided to study DNA chemistry systematically. His analysis of the base composition of DNA from different species produced the specific observation, published in 1950, that has become known as Chargaff's rules. The amount of adenine in any DNA sample always equals the amount of thymine, and the amount of guanine always equals the amount of cytosine. These ratios were constant within a species but varied between species, implying that the specific sequence of bases in DNA was characteristic of each organism. Chargaff's rules were the chemical clue that base pairing was the key to DNA's structure, that adenine always paired with thymine and guanine always paired with cytosine, and they were the specific piece of chemical evidence that Watson and Crick needed to complete their model. When Watson and Crick met Chargaff in 1952 and he explained his rules, they had not heard of them. The specific embarrassment of 2 researchers working to solve the structure of DNA being unaware of the most important piece of chemical data available about DNA's composition tells you something about both the specific information environment of British structural biology in the early 1950s and the specific intellectual confidence of the 2 Cambridge researchers that made that ignorance possible. Rosalind Franklin was a physical chemist and X-ray crystallographer who joined the Medical Research Council unit at King's College London in 1951, specifically to work on the X-ray analysis of DNA. She was, by the consensus of those who knew her work and by the evidence of the photographs she produced, the most skilled X-ray crystallographer working on DNA. Her technical precision and her systematic approach to the crystallographic analysis of the molecule produced images of such clarity and information content that they constituted the most direct available evidence about DNA's structure. Her photograph 51, taken in May 1952, the clearest X-ray diffraction image of the B form of DNA ever produced, showed, to anyone who understood X-ray crystallography, the specific helical structure of the molecule and the specific dimensions of the helix with a clarity that no previous image had approached. The specific measurements that could be extracted from the photograph, the repeat distance of the helix, the angle of the helix, the spacing of the base pairs, were exactly the data that a structural model needed to match. Franklin had not published this photograph. She was analyzing it carefully as part of her systematic approach to the crystallographic problem. not yet ready to make structural proposals before the data supported them. But in January 1953, without her knowledge or permission, photograph 51 was shown to James Watson by Maurice Wilkins, Franklin's colleague at King's College with whom her working relationship was deeply dysfunctional, during Watson's visit to the King's College laboratory. Watson was also shown, without Franklin's knowledge, a summary of her unpublished research data that had been included in a Medical Research Council report, which provided him with the specific unit cell dimensions of the B form of DNA that he and Crick needed to verify their structural model. Watson looked at photograph 51 and understood immediately what it showed. He described his reaction in his memoir The Double-Helix, published in 1968 and notorious for its specific portrayal of Franklin, as his pulse beginning to race. He had seen the clearest evidence for the helical structure of DNA ever produced. He returned to Cambridge and told Crick. They began building the model. The Cambridge competition. Watson and Crick were at the Cavendish Laboratory at Cambridge University. Watson, a 23-year-old American postdoctoral researcher whose previous work had been in bacteriophage genetics, Crick, a 35-year-old British graduate student whose physics background and whose specific genius for theoretical structural reasoning made him the ideal partner for the model-building approach. They were not officially working on DNA. Their supervisor, Lawrence Bragg, had at one point actually ordered them to stop working on it because of an agreement between the Cavendish and King's College that DNA was King's territory. They continued anyway. Their method was model building, the specific approach of constructing physical models of the molecular structure and testing them against the available experimental data, an approach pioneered by Linus Pauling, who had used it to determine the alpha helix structure of proteins. Watson and Crick built their models from metal plates cut to the shapes of the component molecules and held together with clamps and wires, a system more reminiscent of a child's construction toy than a cutting-edge scientific instrument, but whose specific three-dimensional physical reality allowed the testing of structural hypotheses in ways that mathematical analysis alone could not match. The first model, constructed in 1951 after Watson heard Franklin present her crystallographic data at a seminar in London and returned to Cambridge with a misremembered version of her reported unit cell dimensions, was wrong. Franklin told them it was wrong. They were instructed to stop. The second model, constructed in February 1953, after Watson had seen photograph 51 and the MRC report with Franklin's unpublished data, and after Chargaff's base pairing rules had been incorporated, was right. The specific moment of the double helix's solution is one of the most celebrated in the history of science. Watson arrived at the laboratory one morning and began working with cardboard cutouts of the 4 DNA bases, arranging and rearranging them on his desk, trying to find the base pairing arrangement that satisfied both the crystallographic data and the chemical constraints of the molecular structures. He found that adenine paired with thymine and guanine paired with cytosine in a specific geometry that allowed the 2 strands of the helix to be complementary, that each strand was the mirror image of the other, allowing each strand to serve as the template for synthesizing the other. He ran to find Crick. Crick, examining the model, confirmed that it worked, that the structure satisfied all the available data and that the specific feature of complementary base pairing implied an obvious mechanism for DNA replication. The double helix could unzip down the middle, each strand serving as a template for the synthesis of a new complementary strand, producing 2 daughter helices each identical to the parent. The mechanism of hereditary transmission was written into the structure of the molecule. The famous words that Crick spoke to the patrons of the Eagle pub in Cambridge that lunchtime, "We have found the secret of life," were not, by the standards of 20th century scientific modesty, an exaggeration. The paper was submitted to Nature and published on April 25, 1953, 868 words, one figure drawn by Crick's wife Odile, the understatement of the century in its final sentence. It has not escaped our notice that the specific pairing we have postulated immediately suggests a possible copying mechanism for the genetic material. The specific British scientific modesty of describing the mechanism of hereditary transmission as a possible copying mechanism stands as one of the most perfectly calibrated understatements in the history of scientific communication. In the same issue of Nature, Franklin published her own crystallographic paper and Wilkins published his, the 2 King's papers providing the experimental support for the Watson-Crick structural model. Franklin's paper contained the crystallographic data that was the primary experimental evidence for the helical structure. The specific relationship between the three papers, Franklin's experimental evidence, Watson, and Crick's structural model, was not fully acknowledged in the paper itself or in the Nobel Prize citation that followed. The Nobel Prize in Physiology or Medicine was awarded in 1962 to Watson, Crick, and Wilkins. Rosalind Franklin had died in 1958, of ovarian cancer, aged 37, the specific irony that the woman whose X-ray photographs provided the crucial data for the double-helix structure died 4 years before the Nobel Prize was awarded, and Nobel Prizes are not awarded posthumously. The specific question of what Franklin would have discovered had she been given the time, whether she would have solved the structure herself, whether she would have shared the Nobel Prize, is one of the most intensely debated counterfactuals in the history of science. The evidence of her systematic approach and her analytical rigor suggests that she was very close to the structural solution in early 1953. The specific disadvantage of her position, working in an institution whose dysfunctional interpersonal dynamics had disrupted the collaboration that might have accelerated her analysis, without the theoretical structural chemistry background that Crick brought to the Cambridge Group, and without knowledge that her data was being used by the Cambridge Group, may have been the specific set of circumstances that allowed Watson and Crick to publish first. The world after the double helix. The specific transformation that the Watson-Crick structure produced in the life sciences is difficult to overstate. Within a decade of the 1953 paper, the genetic code, the specific mapping between DNA base sequences and amino acid sequences in proteins, had been worked out, primarily by Marshall Nienberg and Hargobind Khorana. The central dogma of molecular biology, that genetic information flows from DNA to RNA to protein, was formulated by Crick in 1958 and has provided the organizing framework for molecular biology ever since. The PCR technique, the polymerase chain reaction developed by Kary Mullis in 1983, which allows specific sequences of DNA to be amplified billions of times from tiny samples, is the foundation of modern forensic DNA analysis, medical genetic testing, paternity testing, and pandemic response diagnostics. The Human Genome Project, the 15-year international effort to sequence all 3 billion base pairs of the human genome, completed in 2003, is the most ambitious biological project in the history of science. CRISPR-Cas9, the gene editing technology developed in 2012 by Jennifer Doudna and Emmanuelle Charpentier, which allows specific sequences in the genome of any organism to be edited with unprecedented precision, is already being used in clinical trials for genetic diseases and has the potential to transform medicine in the 21st century. All of it traces back to the double helix. All of it is built on the specific structure that Watson and Crick proposed on February 28, 1953, based on data that included Rosalind Franklin's photograph 51 and Erwin Chargaff's base pairing rules and the decades of biochemical and genetic work that preceded them. The molecule has been there since the beginning of life on Earth, 3.5 billion years of replication, mutation, and selection, all encoded in the same 4-letter alphabet of adenine, thymine, guanine, and cytosine. Every living thing that has ever lived has carried it. Every living thing alive today carries it. The specific sequence that makes you is different from the specific sequence that makes every other human being who has ever lived, and yet the molecule is the same molecule, the code is the same code. The double helix unzips and replicates in your cells today exactly as it has been unzipping and replicating in the cells of every living thing since the first self-replicating molecule appeared in the primordial ocean 3.5 billion years ago. We found it in 1953. In a Cambridge laboratory. Built from tin plates and wire. Based on a photograph taken by a woman whose name was not on the Nobel Prize. The secret of life. They were right. That is exactly what it was.